comprehensive mathematical framework for computational biology, Dynamical Graph Grammars, which you see here for cytoskeleton in plant cells and synapses. The 2018 project was led by Olivier Helmont, and you could think of this as a kind of spin-off on the modeling side, which is work in developing this framework for modeling very structured kinds of and dynamic biological systems, such as developing tissues made out of cells that are adjacent to each other in a graph, and the adjacencies change as the cells divide and so on. But here, the example has to do with cytoskeleton, with the cortical microtubule array, which you heard about recently yesterday in a plant cell. I don't know if you can hit the little X that gets rid of that. Thanks. Some of the characteristic things that happen, as you can see in these confocal image sequences, are illustrated in cartoon form here, where when one cortical microtubule hits the side of another, there are three possible, well, first of all, they treadmill, they move along, and that's the first rule here. You can have a leading edge of a microtubule turn into an interior node, followed by a new leading edge with a certain probability per unit time, and the reverse. And then when you have a collision, there's three outcomes. One is bundling, if the angle is shallow. One is catastrophic retraction of the collider, and the other is sort of jumping over and forming a junction. And these are all naturally represented by these graph rewrite rules, where you have a pre-event graph and a post-event graph, and various parameters like positions and orientations, and a probability per unit time function that tells you what are the chances of this happening, and if it happens, what are the post-event parameters. And so this is one kind of rule, and the other kind of rule is rules that don't change the structure but do have a differential equation attached, so that you can have dynamics, for example, ordinary differential equations, and potentially other kinds like stochastic or partial differential equations. So it's a very generic framework, particularly for complex biological systems, and can describe them at various different levels of scales of resolution, and even potentially provide the wherewithal to model changes of scale of description. So here's the plant example. We have a new simulator thanks to Eric Medvedev, and so it deals with, here's a disordered system, and here's a more ordered phase in the latest and greatest C++ simulator developed by Eric. Before that, we were running in Mathematica, which is more expressive but slower. It's also been applied by Matthew Herr and previous graduate students to the dendritic spine head, the actin network, where you have a collection of rules like this that alter the structure, and an equal number of rules that describe the biomechanics of these actin fibers as they get pushed and pulled and collide with the membrane and get experience and generate pushes, both push the membrane and get pushed by the membrane. So if you're interested in this, then you can come and talk to me about the details, and question about, is this closed under model reduction, how expressive is it, at poster 4 in the Wednesday's poster session. Thanks.
Thank you very much.